All right, so we'll pick up here with part two of our cell lecture. We'll start by talking about this structure called the glycocalyx. And so, what glycocalyx is, if you think of the word glyco, we're referring to glucose, we're referring to carbohydrates. And so, what this glycocalyx is this outer coating, this outer surface feature of our cells on the surface of the plasma membrane that will consist of varying carbohydrate containing proteins. So, you have glycolipids, so um, carbohydrates attached to a protein. Glycolipids are carbohydrates attached to lipids. Glycoproteins are carbohydrates attached to different protein molecules. And what this glycocalyx does, and I'll show you a picture in the next slide, it serves as essentially an identity tag or something that our immune system and the rest of our body can look at and sense and be able to tell, okay, this is normal. This is self. It is unique to every person, and this can be important in different types of, if you're having to transfuse different things or an organ transplant, you need to make sure that they are compatible between, and part of that is due to this glycocalyx. And so you can look here on the luminal surface of this blood vessel, you can kind of see this surface lining here that kind of looks slightly hairy, if you will, and you can see the same thing here. So what you have is you have your lining of your blood vessel, so an endothelial cell, and then projecting off its surface is this glycocalyx. So this is what is being shown to the rest of our body, shown to the immune system, that this is a self cell, is unique to this particular individual. And remember, again, it's made up of glycoproteins and glycolipids. So on the on the apical surface of our cells, roughly in the location where the glycocalyx is, we have different specializations that we can see. We have one called microvilli, and all microvilli are these convolutions and these twists of our plasma membrane, these extensions that serve largely to increase surface area. And so, if you think about why or where you would want to increase surface area, you can think maybe we're wanting to increase surface area so we can contact more of something. And this is exactly what we see in our small intestine when we're talking about absorption. We have microvilli. We also see this in parts of the kidney as well, in the proximal tubule. We are increasing surface area so we can contact more things that we have just digested, if we're talking about the small intestine. And we can then bring those things into the cell because we're contacting more of it because we have more surface area. Okay, same concept goes and is true with our kidney. In our proximal tubules in our kidney, we have microvilli that serve to increase reabsorption of water and other solutes within our kidney. Okay, now connecting our cells together, we have varying cell junctions. Okay, so we have things that are tight junctions, we have things called desmosomes, and we have another one called gap junctions. Okay, so tight junctions, if you think about the name tight junction, it is going to very closely and almost completely seal the cell. Okay, sometimes they are arranged in this kind of zipper-like fashion around cells that hold them very tightly together to prevent things from getting through. However, a lot of tight junctions are what you can call selectively leaky. So meaning that they will selectively allow very certain things to pass through, but not others. Okay, so they're largely serving to keep things together. We also have desmosomes. Desmosomes here, sometimes they're also called macula adherens, you'll see them. They will exist between two adjacent cells, and they look something like a spot weld. They look like this little dot, and then in between these two dots, within this kind of space between cells, you have these proteins called cadherins, and these cadherins will stretch across and adhere to one another and keep the two cells very closely joined together. Okay, and then lastly, our gap junction that you can see over here to the right. A gap junction is exactly what it sounds like. It's a gap between two cells The structure itself is called a connexon, and a connexon is made of multiple smaller molecules and proteins called connexins. So connexon is the connexon, and then connexin, connexin, are the individual proteins that make up the connexon. Okay, and gap junctions primarily they serve to allow for communication between cells, and that makes sense because it's an opening. It's this confluent space between two cells that would allow ions or solutes, or in the event of cardiac muscles, you can also think of electrical signals can be sent from one cell to another because of these gap junctions. In cardiac muscle cells, you have something called an intercalated disc, and within that disc, you have a couple of things. You have desmosomes. You also have gap junctions, and because of these gap junctions, it allows the current, this electrical flow flowing throughout the heart, to pass from one cell to another very quickly. So here's a picture of a desmosome. So you can see these little spots here, these spot welds. Sometimes they're called plaques. And then you have these cadherin molecules that stretch across and keep the two cells closely joined. You also have something called a hemidesmosome. And instead of joining one cell to its adjacent cell, a hemidesmosome is generally on the basal surface or the bottom surface of a cell, connecting the cell itself down to the basement membrane to keep it somewhat adhered and linked to that. Here's another picture of desmosomes here. You can see these cadherins as they stretch across this kind of intercellular space. Here you see tight junctions. Here they kind of form this zipper-like network around the cell that are keeping them very closely adhered, and for the most part preventing any type of passage of solutes or any other material. And then here we have a gap junction. So what you see is a connexon, this one large unit here, and each of these little individual pieces, these are all connexins. So multiple connexins come together to make a connexon. Okay. So we will now talk about cell permeability and transport. So we talked a little bit about with these different proteins here, the fact that some of these will allow things to cross the cell, some will not allow them to. And so this idea of cells being permeable refers to the fact that some things can, some things can't. Okay, it's selectively permeable. So when we're talking about this, only certain things can pass through. So what may be moving is a solvent or something that something else is dissolved in, like water, or a solute, which would be the substance that's dissolved in the solvent. So a good example to give is think of a glass of water and then think of salt. So you have a glass of water in one hand and you have a lot of salt in another hand. The water is your solvent, okay? Your salt is your solute. You can put the solute into the water, and now you've dissolved your solute, which is your salt, in your water, which is your solvent. Okay, so that's a nice example, or a nice way of thinking about these two things. Now, the way things move within cells and out of cells is they move from an area of high concentration to low concentration. Generally, let me go back. They generally move in that direction, and they move in that direction without the use of energy. Okay, it is a passive process. It's called moving down its concentration gradient. You're moving from somewhere where it's highly packed to somewhere where it's less packed. So you can think of this if people were in a room. Lots of people were in a very crowded room, or a very small room. So if you have lots of people in a room, the tendency if somebody opens the door is for people to start moving out. And so they're moving from where they're highly concentrated to somewhere where it's less concentrated. That's this idea of passive transport or diffusion, moving down. Now, you can have the exact opposite, an area of low concentration to high concentration. But what you may be thinking is that this is not as easy, it's not as intuitive as moving from something high to low. And so what this is called is active transport. And what we mean when we say active is that it requires energy. Okay, passive because it does not require energy. We'll then see that between passive and active transport, there are a couple of variations of each one, and we'll talk about what those are. So, passive transport. Our first example, like I said, no energy required, completely free of energy. Simple diffusion. This is simply things moving from high to low. We don't require any type of protein to help us get there. We're just moving down. Understand, though, that although things are moving from high to low, there's also a little bit of whatever we're talking about, if it's a particular solute, or think of like sodium or potassium. There's also a little bit always moving the opposite direction. So most is moving high to low. Some of it will move from low to high, back and forth. Okay, but the idea is that in general, overall, we end up at equilibrium. So we end up at a state of homeostasis and a state of equilibrium. If you're talking about maybe two sides of a cell, an extracellular space and intracellular space will always end up at equilibrium. Okay, so that's simple diffusion. No proteins required, no energy required. Now, we can have facilitated diffusion. It's still passive, it still does not require energy, but now we have to use a protein to help us. Okay, we see this sometimes whenever we're transporting amino acids or glucose into or out of a cell. We require a specialized protein carrier to do this, so-called carrier-mediated transport, but it's still completely passive. We don't require energy. And then lastly, osmosis. I think there's actually another one, but after osmosis. Osmosis is the process of water diffusion. So water is moving from an area of high concentration to low concentration. And what was discovered, and it was actually the man named Dr. Peter Augrey at the Johns Hopkins University. He and his research team discovered the aquaporin channel, and there's now been it's now confirmed that there are 11 different isomers of aquaporin channels, or 11 or so. Different ones function slightly differently, but what's happening here is that water is moving through these channels. It's the water channel of the cell. It's essentially the plumbing system, and so things are able to move from one area of a cell to another through these aquaporin channels. Okay. Now, our last one is filtration. We see aquaporin channels in this process of osmosis in our kidney, where we're also talking about filtration. But filtration, and what we mean here specifically, is the movement of molecules and solutes and things out of a fluid due to the pressure of that fluid, so due to the pressure of the water. So when you talk about glomerular filtration in the nephron, you talk about hydrostatic pressure and you talk about osmotic pressure, or colloid osmotic pressure. These two are opposing each other, but hydrostatic pressure is the pressure due to the presence of water pushing on a particular surface that is causing some things, like some type of molecule or solute, to leave that mixture, to leave that water, that liquid. And so we see this in the kidney, in the Bowman's capsule within the glomerulus. So that is all active transport. None of what we just talked about requires any type of energy. It's completely passive. However, some, like facilitated diffusion, it does require a carrier protein. Now, active transport, on the other hand, is now going to require ATP. We're now moving things from an area of low concentration to an area of high concentration. So it's not as easy to do that, so we have to put energy into the system, okay, in the form of ATP. So we have primary active transport and we have secondary active transport. We'll talk first about primary. So primary is you're using ATP directly via a protein to transport something across the cell membrane, across the plasma membrane. A good example of this is our sodium-potassium pump, or our sodium-potassium ATPase pump. What it's doing via ATP, via the hydrolysis of an ATP, it is moving 3 sodium ions out of the cell, so to the extracellular space, and is moving 2 potassium into the cell, or into the intracellular space. And what you see here is you have hydrolysis of a triphosphate, hydrolysis of an ATP. you get ADP and an inorganic phosphate. That energy release is enough to power this protein, this ATPase, to pump 3 sodiums out and bring 2 potassiums in. So this becomes very important when you're talking about action potentials, because action potentials require a polarization of a membrane. There has to be a difference in charge. And in order to keep your difference of charge, you have to have certain ions and certain solutes out of the cell, and you have to have certain ones inside of the cell. So that's primary active transport. Use of ATP directly, in an example is our sodium potassium pump. Now, secondary active transport, it's requiring energy, but not directly using ATP. What it does is it uses the energy stored in another solutes or another molecule's concentration gradient. So something has to be moving from high to low, which you generally think of as passive transport, but then that energy used there in that process is enough to allows enough energy to pump something else either in the opposite direction or also in the same direction. If it's pumping something in the same direction, it's going to be called a symporter. If it's pumping something in the opposite direction, it's going to be called an antiporter. Okay, but both of these are types of secondary active transport. So, sodium glucose transporter, this is going to move 2 things in the same direction. So, in order to get glucose into the cell, it will ride alongside sodium as it's coming in. You can also have an antiporter, a sodium hydrogen antiporter. You see these, I believe, in the kidney. If you are wanting to reabsorb, if you're wanting to get rid of hydrogen to help with maybe acidity or pH levels, you can get rid of that hydrogen by also bringing in and reabsorbing a sodium. Okay, so you have to look at, are we moving in the same direction or are we moving in the opposite direction? Okay, all right. We will stop there and we'll pick up with vesicular transport in the next part.